Hello, and welcome to this webinar titled Efficiency by Design, Case Study for a Hybrid Clinical Manufacturing Facility, hosted by Biopharma Asia magazine and presented by Dr. Sourav Kundu, Senior Director of Process Development and Clinical Manufacturing at Teva Pharmaceuticals, and Adam Kaletski, Business Leader of Bioprocess Hardware and Enterprise Solutions at GE Healthcare. My name is Stephen Edwards, and I will be your moderator. Now allow me to introduce our first presenter. Sourav Kundu, PhD, is a Senior Director at Teva Pharmaceuticals Global Biologics R&D. He directs the development team involved in cell culture process development from clone to commercial. He also heads the clinical manufacturing for biologics at Teva. Sourav has over 25 years of industry experience and has been involved in development and commercialization of multiple biologic drugs. Prior to Teva, Sourav was a Director of Process Development at Amgen. He also held positions with increasing responsibilities at Aventis and Baxter. I will now be handing over to our first presenter. Welcome, Sourav.
Thank you, Stephen, and welcome to all of you that have joined the webinar. I would like to thank BioAsia for organizing this event and inviting me to deliver this webinar. And also thanks to Adam for being a co-presenter with me. Today I'll be talking about how efficiency can be built in when designing a clinical manufacturing facility by combining in a very strategic way single-use and stainless steel systems, and thereby creating a hybrid manufacturing facility. So I'll go to the second slide now. Typically, hybrid manufacturing facilities combine stainless steel and single-use systems. And there are many stainless steel plants with some single-use equipment incorporated, and that is done mainly to handle small volume media or buffer storage using single-use bags, things like capsule filters, pod-style filters that are single-use, and small culture vessels and wave-type bioreactors that are also single-use. These add flexibility to the facility and also help reduce bottlenecks with typically with media or buffer tank availability and intermediate pool storage. So in our case, we actually defined hybrid in a little different manner. We said that the hybrid is mostly single-use equipment in combination with some stainless steel equipment where it makes strategic sense. We all know now that the single-use systems offer flexibility. They can be designed in a very modular and adaptable manner. They are quicker and cheaper to validate. They're easier to operate. Because of the single-use nature, there's no CIP, SIP requirements, and that gives us an advantage for water usage. And it has also been demonstrated now over time that the single-use systems actually can offer a higher success rate, and that's because the single-use systems are pre-sterilized, so the risks associated with typically with CIP, SIP systems are no longer present when we're using single-use systems. On the other hand, stainless steel equipment can provide some advantages as well. Sometimes they're technologically suitable or superior, particularly when you think of something like a centrifuge, which is a complex machinery Stainless steel has demonstrated its technological superiority. They can also be more reliable in some cases than single-use. Obviously, they have proven track record, and sometimes they can give us a better process fit. Now, whether a company decides to use all single-use, all stainless steel, or maybe a combination of the two, that's a decision that typically has to be made by the company based on its goals, objectives, and needs. And there are camps on both sides. So some are going completely single-use, and some have decided to, still stay with stainless steel, depending on their business objectives and goals. Okay. Oh, I probably have clicked extra time. So, okay. Yeah, that's the correct slide. All right. So we're now going to go back to our clinical manufacturing facility. And the decision for us was to go with primarily single-use systems with the key pieces of stainless steel equipment that was driven by a few important considerations. We wanted to maximize the flexibility to accommodate new processes, evolving process technologies. This is a clinical manufacturing facility, so by definition, we have to be able to accommodate different types of processes and also process technologies. We wanted to minimize the timeline and cost for building this facility. We were working with an existing building, so we had to fit everything within the existing space. We wanted to also not go through a lot of complicated validation, so all the validation and qualification requirements needed to be minimized. We wanted to add operational efficiency and increase success. So we incorporated stainless steel equipment where it really made strategic sense, and it supported the process requirements. And always, for a clinical manufacturing facility, it's important to build for future unknowns. So we're now going to go into a little bit deeper into the case study. So this is the hybrid clinical manufacturing facility for Teva Pharmaceuticals, and the goal for the company for building this was really to advance our biologics pipeline. So it was determined to be a very strategic asset for the company that would enable us to go very rapidly from bench to clinic. We wanted to build a one-stop shop for clinical manufacturing, so the facility actually has everything from cell banking to drug product fill finish. So we don't have to rely on CMOs. We could just have everything under our control. The facility is about 19,000 square foot, fully GMP compliant, all regulations, worldwide jurisdictions. It's supported by a QC lab as well as a material warehouse, and the utility spaces are right next door within the same building for supplying water for injection, gases, and so on. So it's a fully integrated, fully functional manufacturing facility. We used an existing shell space. So the building was actually existing. It was being used for a different purpose, so we had to repurpose this building and gut the inside and build everything new. However, if you look at the picture on the right, you can see that there are lots of odd corners and angles and so on that posed really big challenge for us for laying out a nice logical manufacturing facility. We also had some structural limitations in terms of how much weight can we put on the roof, and also some height restrictions with respect to ceiling height and the height of the interstitial spaces. However, at the end of the day, despite all the challenges, the design team was able to provide a really nice and operationally efficient facility layout. The facility, and you can see kind of like a pictorial representation of the different rooms here. Picture's a little bit small to see, so I'm not going to go through in detail. But basically, it goes from the locker rooms from one side to the processing rooms to the other side, and the utility spaces are also adjoining the manufacturing area, so that allowed us to have shorter pipe runs for utilities and save energy and cost. The facility has a unidirectional personnel flow, so people go in through the locker rooms, go into the supply corridor, go into the manufacturing processing rooms, and then come out of the other side and take the return corridor to exit the facility. Has a wraparound hallway, and there are some nice room adjacencies that we're able to achieve here. So for example, if you look at the picture, the media prep and cell culture are right next door to each other and just separated by a wall. The wall has some ports built in that can be opened and closed. So what we do is, after we prepare medium, we pass the hoses through those ports, which then connect to the bioreactors on the other side of the wall. It eliminates moving these single-use mixers back and forth between rooms, helps us with our ergonomics and really hassle. So same type of concept between buffer prep and purification rooms, which has access to both processing rooms through the ports built into the walls. The facility has full viral segregation, so we actually have pre-viral and post-viral, where the post-viral is a grade B area. The cell banking is a fully self-sufficient grade B banking facility, and then the drug product manufacturing facility, which is at the very end of the building, also fully segregated from the rest of the facility. So this type of design actually allowed us to create everything from soup to nuts under one roof, and maintain all of the requirements, regulatory requirements and expectations. Here, this slide shows a little more details of the types of process equipment. So we try to cover a broad range of process types and scale. So we have bioreactors that are 50 to 2,000 liters, single-use bioreactors. They allow using different types of seed train strategies and a fairly broad production range. The harvest, we kept the ability to centrifuge because of the higher cell densities, et cetera, that we're achieving these days. So we wanted to maintain that capability, not that we needed to use it for every process, but we just wanted to have that capability. And we also have, obviously, pod and Kuno depth filter systems that are typically our workhorse for harvest. On the downstream side, we decided to stay with the more traditional stainless steel active process skids, and primarily that was because we needed the flow rate ranges that those stainless steel skids can offer. We have Axichrome columns for different sizes, and they're really nice columns. They've been really good in terms of the reliability for packing. We have two different UFDF skids, so depending on the amount of processing that we have to do or the concentration that we have to hit. And the media buffer prep is all single-use mixers and totes, and very varying volumes to meet the process needs. On this slide, I'm going to talk about a few project organization and structure that we utilize to be quick, efficient, and really have streamlined decision-making steps. On the right-hand side, I'm showing a few pictures. Pictures are worth way more than the words. And you can see this is sort of a time sequence photograph. So you can see on the top, when the inside was just a shell space, and then gradually things started to appear, and now it's actually a little too full than we would like. So the capital project team was actually led by what we would call a core team. They were empowered. They represented all major functions like manufacturing, engineering, quality, process development. And the people that actually led the core team and the representatives were the decision makers and people that will actually be living with the consequences of those decisions. So it was to their interest to make the best decision that was possible. And we had a fully integrated schedule that allowed strict oversight of all the activities. The engineering was outsourced to an A&E organization, and we also ended up awarding the construction to a separate company that was not connected to the design. And it actually worked out really well for us because we had a second set of eyes that looked over the design, and we did find a few things that we needed to adjust. The subcontractors that this construction manager brought in were all very, very well known in the industry with lots of experience. It did require some strong negotiation with the procurement groups, but at the end of the day, I think we were all better off. And we were able to achieve shell to substantial completion in about 10 months, which is really, it can be done with single-use systems. And we're really happy to have that. We used a very streamlined qualification strategy, and that focused on risk, priority, and it was totally based on common sense. We had a very small internal validation team that was basically managing all the qualification work that were being supported by contractors. We did a lot of risk assessments and harmonized URSs with the vendors. We used extensively the methodology recommended by ASTM E2500, where all of the qualification activities were based on risk. And in many, many cases, we leveraged vendor testing upfront, for example, during the FAT, to eliminate redundant activities once the equipment was brought in and installed. We purchased commissioning and qualification as much as possible from the vendors, and we were able to complete all of the qualification work in about 5 months, and which allowed going straight to shakedown and being able to start campaigns out of this facility. Because we were very close and closely working and functioning team, we were able to handle the startup challenges and quickly resolve them. We had the typical startup issues, some mechanical parts that did not work up front, and some electronics that didn't work. There were some mismatched components, some out-of-box failures, some automation code issues, so very typical in any kind of a startup of a facility. Had some issues with the Wi-Fi system, although they were very quickly resolved, and just with some simple changes in the sanitization scheme. And what was really important was that there were no changes necessary to the design of the equipment, or we never really had to go and buy a new piece of equipment because we didn't select the right equipment. So that worked out very well for us. Okay, so this is sort of a slide to summarize what we just talked about. We just talked about a hybrid manufacturing facility that was designed and built to enable an emerging biologics pipeline. We designed stainless steel equipment, or I should say incorporated stainless steel equipment in a mostly single-use facility, and that was done for strategic advantages, primarily driven by technology, track record, and the ability to match process requirements. The single-use systems provided higher flexibility, greater operational efficiency, nice modular design, and low initial cost and shorter timeline for installation and qualification. We were able to simplify our CQV strategy, and that was possible as a result of leveraging vendors' internal testing and using a fully risk-based approach. We were able to quickly resolve our startup challenges and move to a successful PQ. And at this point, so this slide is a bit dated, but at this point, the shakedown obviously has been completed with 100% success. Shakedown allowed us to operate all equipment, automation, and utilities, and make sure that everything was in working order. Since this time, we have now completed 2 additional campaigns, and everything seems to be going well. So I'm going to finish by showing you a few pictures, and there's a little lag here, so bear with me. And the pictures are always worth more than the words, so I figured I give you a snapshot of what the facility and the rooms look like once everything was said and done and people put in their hard work. So here you can see some of on the top left, the bioreactor room, and on the bottom is the purification room with the active skids that we talked about. You can see here in the middle the adjacencies of the rooms that we talked about. Everything is very open. There are lots of windows so that people can see what's going on in the next room. So that allows, more efficient and more streamlined manufacturing operation. I think this picture was taken in the, maybe this is the buffer prep area. You see some utility equipment in the utility space, and also all the air handlers, etc., in the back of the room. So as you would expect in a manufacturing facility, there are lots of big equipment. A lot of them are utility equipment, takes up a lot of space, but they all have a purpose, and that is to make sure that we achieve our business objectives, and we ultimately, at the end of the day, we can help our patients. So this is where I'm going to stop and hand over to Adam.
Thank you, Sourav. I'll just interject. Once again, I just wanted to remind our viewers that there will be a live Q&A session at the end of the webinar. But please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen. And we'll go through these at the end of the webinar. Now, please allow me to introduce our second speaker. Adam Kaletski leads the Bioprocess hardware business at GE Healthcare in North America. He directs a team of scientists and engineers that deliver solutions to help customers solve their development and manufacturing challenges. Adam has been designing systems and technologies in numerous industries for over 30 years. He has spent the last 23 years with GE Healthcare, delivering innovative solutions for the pharmaceutical and biotech industry. He holds several patents around biomanufacturing technologies and holds a Bachelor of Engineering in Electrical Engineering and Computer Science from Stevens Institute of Technology. I will now be handing over to our second presenter. Welcome, Adam.
Thank you, Stephen, and thank you, Sourav, for a great presentation. I really appreciate the opportunity to present for you here. So, Bioprocess Asia, thank you for your attendance as well at this webinar to all the attendees. We're going to talk a little bit about practical considerations in hybrid manufacturing. As you saw, the great solution that Sourav and his team have put together at Teva, I think they did a great job with that, and it's pretty well established today in the industry about traditional technology, stainless steel and cleanable. So a bit of my presentation is going to focus on single use, which is an area that less familiarity exists on. So we'll cover a number of topics. We'll look at market dynamics that kind of talk about why the single use trend seems to be happening. A little bit about the economics of single use versus stainless steel. Hybrid does seem to be the rule right now, but we're seeing more trends towards increasing use of single use technologies. We'll look at the benefits then when we switch over to when we want to implement a parallel production when you're trying to put a single-use facility in place and the timelines that you can save. Talk about why would you want to standardize, because if you standardize, you have a lot of advantages. We'll talk about those in my presentation later on, and then some deciding factors, whether you build or outsource. So the industry, it's been quite a wild ride over the last 20 or 30 years. In this industry, there's been a lot of change. If you could think back to the 90s and the 2000s, prior to that time, pharmaceutical was a small molecule world. It was really dealing with very simple, very large indications. And when biologics came to play, it changed the industry. I mean, it was as game-changing for pharmaceutical and treatment as Uber was for taxis and GPS was for the old mapping world. So it was a huge change, and these biologics made a huge amount of valuable improvement for patients and patients' lives. But like every change, the next generation comes forward and causes a new interruption. And biologics are coming off patent. We've got, as a result of that, it's changing things. The largest indications are all being dealt with, and they're looking for smaller indications. Diverse drug pipelines are coming down as a result of that. And then we're seeing this new trend that could be the Uber for biologics and could completely disrupt. Where we've been dealing with treatments up till now, personalized medicine is showing the possibility of cures, and that's an enormous enormously positive movement. But it's at the early stages right now. The CAR T successes are very exciting, but this is the beginning of the pipeline of personalized medicine. To do this, you're going to have localized manufacturing, and that's what people are looking to do, is manufacture locally. In personalized medicine, it's as local as a single patient. In biologics manufacturing, it might be smaller lines with single use. So it's really important to recognize this evolution, because it creates a set of challenges. These newer drugs are being challenged by healthcare costs, so we have to look at how do we manage the costs on this. We have to adapt quickly. There's all sorts of changes that are happening. If you see a possibility to make a move to, for instance, a biosimilar, you might jump on that quickly, and the first to market has always gone the best in terms of how much money they can make and take from the opportunity. And then finally, the time to market is a part of that. How long it's going to take after you decide you want to move into a market to make that drug? These are all challenges that we have to look at, and hybrid and single-use technologies are ways to move forward on those challenges. Let's look at the landscape of biomanufacturing in the world today and tomorrow. If we look today, the majority of the capacity exists in North America and Europe, Western Europe. So if you look at that, the majority of the people living around the world are actually in the other regions of the world. So today, with 2/3 of the manufacturing capacity in these other areas, you'd expect that with the movement, you'd see growth. And in fact, we do. You can see that the largest capacity exists in those two established markets, but the largest growth is happening in the other developing markets. Biosimilars are causing a lot of this driver, because now it allows for these drugs that would spend so much time being developed to be manufactured quickly and easily with a lot less scrutiny. You'll see that the challenge you have when you start to develop quickly, 20 years of development happen in these developed areas, and you're doing it quickly in these emerging markets. So the high growth, you immediately have a challenge of how do we move quickly to not take 10 years to develop that. Single use and hybrid can give that advantage to the teams. So the dynamic biologics landscape will naturally favor agile manufacturing. If you can speed things up, you're going to get to market quicker and break this bottleneck up that we're seeing here. We talked a little bit about market trends. Let's look further into the future. So in our analyses of the industry, we're thinking that by about 2020, the biologic drug market is going to be about a $390 billion market. The challenge is to meet that capacity, we think we're going to have a capacity of about 73% that we're going to have by 2020. And unfortunately, right today, 50% of the bioreactors are 1,000 liter or smaller. And so these large blockbuster drugs had large bioreactors of 10, 15 or more thousand liters, and single-use bioreactors today, and most bioreactors today, are under 1,000 liters. So that opens up a question of how are we going to meet this demand. And finally, where is it all happening? As I showed you on the previous slide, we think Asia will probably be about 24% of the global capacity by 2021. So a huge shift of where things are developing right now, since the capacity already exists in those traditional markets. What's driving this? Well, if we look at 80% of the drugs in development versus 80% of the infrastructure of drugs that are currently established, the two don't match. They just don't match. The majority of the drugs produced about 100 to 500 kgs per year. The traditional titers that have been developed for these drugs over the last 20 years are in the 1 to 3 grams per liter range. But if you look at the drugs in development, you can see that the drugs in development are much smaller volumes and higher titers. So titers are now reaching the 5 to 10 gram per liter range. So in the same volume, you can have a lot more therapy. And so with that trend of smaller batches to meet things like orphan drugs and smaller indications developed at higher titers, it opens up the door for different technologies. So a hybrid is an approach that most customers are now moving towards, as you heard from Sourav. If you look at a heat map to understand this, you can see that traditional stainless steel has advantages at lower titers and higher product costs. Higher product volumes, excuse me. And single use suddenly comes into play when you start to see higher titers and smaller volumes by the nature of how much volume you want to put into plastic seals, plastic bags and such. So this is the driver that's opening the door for stainless steel transition over to single-use technologies. One of the drivers, at least. If we look at the evolution of the bioproduction technologies, when we start to move into single-use technology, what's next? So if you can imagine single-use technology getting the foothold that it's getting, it's doing so primarily because of the reduced CapEx experienced by that. But it opens up the idea of these smaller unit ops with closed system capabilities, cross-contamination risks that come to play when you don't have to worry about the cleaning validation as strongly. And then, of course, the multi-product facility opportunity. You don't have to design today for the next 30 years, but rather you can be much more flexible. And once you've got that in play, then you can talk about how do we connect these processes together. By having single-use connectivity, you actually have simplified steps to integrate. You can use predefined automation in those steps to put them together in a block-type format. And then you can streamline those platforms, because you could put multiple platforms in play, and you can either produce on multiple platforms the same product, or you can diversify those different steps that you're doing on those different platforms. That then lends itself, when you have this flexibility, to a modular facility concept, where you can actually build the facility in modular pieces like trailers, bring them together and place them anywhere you want in the world in a very simple, streamlined way. Once again, in turn, that opens the idea to say, if I put these streamlined unit op modules in there, could I put it together with a shared campus service concept? So a shared service campus might have dedicated utility things like water, facility services, warehousing, waste management, and then you can actually have the modular facilities all hub and spoking off of the shared campus services. So this is the evolution we can see in the future that's going to speed development up. And it could significantly, as we'll talk about moving forward, speed that development cost by magnitudes. If we look at the concept of a single-use line, Sourav showed a great example of modular, and so you've seen that one. Now we can talk about what if we wanted to go full single-use. How could it be done? And any of these steps you see here could have stainless steel or cleanable systems integrated into it. So there's a lot of flexibility when you do this. But the advantage of when you're talking about these single-use and hybrid manufacturing platform concepts, when you put a platform together, you can easily replicate it. There's lots of flexibility. Consistent liquid connections. You can develop your liquid connection technology that moves between the steps in a very easy way. Sourav mentioned how they feed through the different feed-throughs in the walls, and that's a key component of this, the modularity, separation between walls, and how you can utilize these consistent connections. Automation is critical, because when you put the automation together, you can actually communicate across these unit steps in a very flexible way. You can actually design one facility line one way, and the next one sitting right next to it could be done in a different way, but the same uniform automation. Remote monitoring, you want to make sure what's going on, and the increased batch release efficiency, because you have this consistency across the different steps. Helps in validation and training, because customers, the people have to know how to use these things efficiently, and so the training is really important to have that consistency. You know, you have to design automation in from the start. Automation is a key component. If you can't control and see what you're doing, then you're going to have errors and problems across the way. So we always encourage the automation to be considered from the start of the whole product, the whole process, and every product comes with it a consistency across the automation. So it's really a placeholder to say, don't think about it later, but rather start at the beginning when you think about your automation protocols. So if we look at process economy, what's a better value? I don't think there's a good answer to that. I think the key would be dependent upon what it is you're looking for as your priority. So single use has advantages, and stainless steel has advantages, and hybrid gives you an opportunity to take the advantages of both that meet your specific needs and your specific goals. So I'll show in a few examples here how it's a lot less time for a single-use facility to be put together, and your CapEx cost as well. You can be a much lower CapEx cost on single-use. Your OpEx has potentials to be different between them as well, and your changeover time is a huge difference, because with single-use changeover, a lot of the cleaning and and verification protocols are lower. But you know, you got to also look at your outputs. So there's advantages and disadvantages in all these technologies, and therefore there's no right or wrong answer. So you have to then think about this and decide which technique is the best for our specific situation. They do have advantages and disadvantages, which I want to highlight here, because to understand why single use could provide an advantage, you need to look deeper at the phasing. So at drug development phasing. The primary advantage that single use gives, besides the cleaning and other things that we talked about, is it allows delay because it's a faster creation of your facility. It allows delay of the creation of that facility until further along the drug development process. So if you consider a stainless steel plant is going to take about 4 years to build, you're going to have to start long before your launch. So you might be starting in phase 2. And your drug has a lot of potential to fail as it moves forward through phase 3 and beyond. So if you don't have to build your facility that early in the process, your CapEx savings could be great. And any failures that are going to happen could happen before you build. Single-use facilities could take anywhere from 1.5 to 2 years to build. Therefore, you've actually got this space in between that give you that chance to avoid building if a drug is going to fail. In addition, if you're single use and you do build here and something fails, you can repurpose in a very easy way. So this is the timeline versus CapEx cost graph that really shows that advantage pretty clearly. There's also the idea of volume and titer that have a big play on which way you want to go. So when you consider volume and titer, you'll decide which one is the best from a purely quantitative standpoint. You can look at the cost that it takes. So if we were to pick an annual volume of, let's say, 3,000 kilograms at a 5 gram per liter titer, we can calculate which is the more advantageous if we try to keep all of our other variables consistent with one another. If I look at some assumptions here for this tool, this is a tool, by the way, that we're developing that should be live on our website in the next few months. And then you'll be able to play with it a little bit to kind of get this idea behind which way to go in your own hands. But of our assumption sheet, which you see, we're trying to look at the bioreactor setup, make it consistent between what a stainless steel setup and bioreactor setups might be. And we try to keep everything else constant. And in doing so, it brings us the ability to kind of develop which is the better choice in a simple, big picture way. And then you can deep dive it. And you can see with this a very easy relationship between titer and volume. So in this analysis with 300 kigs and 5 grams per liter, we actually have a 34% savings in single use. So single use is the recommended technology. But as we start to go up in volume, that advantage starts to dissipate. And in fact, when we get to a certain volume at 5 grams per liter, we find that suddenly stainless steel becomes a technology that makes more sense. Now, this is, again, looking at a pure technology of stainless or pure technology of single use. So the hybrid is somewhere in between, and you'd look at the individual steps. But in the same way, if we bring that back down to 300 and we look at the titer, as we go up and down in titer, higher titers favor single use. And as you go down in lower titers, you start to get to the point where stainless steel becomes the advantage. So you can think of the old ways where low titers, high volume, stainless steel was the king, and it still is. It's when you go to higher volume, lower volumes and higher titers that you start to trend towards the single-use technology from a pure volume standpoint. So it's a really interesting analysis which you can do by doing this. And where do the costs come from in the different technologies? Well, in all of these different cases, this tool tends to look at that. In single-use, it's a materials cost because you're replacing single-use technologies. In stainless steel, it's a labor cost because you're doing a lot more cleaning, a lot more changeovers and such. And then the other pieces in between are similar, very similar in many ways. So it's a really interesting way to look at how you choose which is the right technology. And again, when this tool comes online, you guys can play with it yourself. So keep an eye on our website and feel free to browse for it. So that's one way to think about which is a better way to go. There's advantages and disadvantages to each of those. But if we look, generally speaking, it's about single use, since this is kind of less known by most people. Again, we have up to 60% faster to market if you install a single-use facility, particularly a modular single-use facility. You could have a 5 times quicker batch changeover because of lower cleaning times and validation. A smaller footprint could be possible because of the smaller device sizes and connective technologies, more flexibility, and a 30% OPEX savings because of the cleaning labor is what we're seeing on this whole thing. So let's think now about if we have a facility. You saw a great example of when you utilize existing technology that Sourav gave, you can actually put a great solution in place with a hybrid solution was what worked best for them. And some of the advantages of those different pieces, like the Axichrome columns he mentioned, are very, very wide range of capabilities and materials. If you wanted to say now, what if we wanted to do a modular facility in a complete design? What we're seeing is a trend now especially in new greenfield build-outs, the idea of analyzing whether a modular facility would be the right choice if you don't have an existing infrastructure. So a modular facility provides a few advantages. It pre-designs things, so your infrastructure is built in a factory to your specifications, but built in a factory. The design is pre-designed from start to finish. It can be easily reconfigured and put together so that when it's time to build it on your site, everything is already built to your specs, to your requirements in the workflow that you need. The workflow has already been pre-developed in many facilities. So if you wanted to go with a traditional biologics facility, for instance, the designs are done in many of these cases. So you can actually pick that design, go with it, and drop it in place, really reducing the cost. And the fabrication, it's easy to develop consistent factories. You can put one in 3 different locations around the world, and it's going to be 100% equal to one another. Management is easy because the project manager has a really good handle on what all the different components of the project would be, and they can do parallel processing of a lot of those different components. Plus, the training is easier because you actually have the training and the pricing all known from the very beginning because it's gone through already. You can see the compression of parallel operations in a facility, where in many stick-build facilities, you actually have to complete one step before starting another. There's a lot more parallelism between a modular facility, reducing the flow by almost half of that project. 18 to 24 months we build these things in nowadays, so you can actually see everything from the planning, right at the part of planning and site permitting, we're actually already building things because we actually have the ability to know exactly what's going to be done in there. So this is a way where you can actually utilize this single-use or hybrid technology in a full-blown facility manufacturing project. And then, as Sourav had mentioned, in terms of other parts of it, you have to do your validation, you have to do your PD work and such. That's another thing that if you can utilize offloading in services that your provider can offer. Like in our case, we have our fast track manufacturing services that can help do some of that work if you needed that help for PD, validation services and such. And then you just plug that right into the facility. That can be done in parallel while the facility is being built, once again, bringing it down to an 18 to 24 month build. So it's a really interesting way to go forward. And by doing so, you can do the work once. And if you're looking to if you're either a global company that has facilities around the world, or you're looking to make moves in different areas of the world, it's an easy way to have capacity managed very well. By the standardized facilities, these companies are able to actually build in any different company, any different group in any different place in the world where they have capacity, which makes it very flexible. This way you can decide we're going to repurpose facility number 2 to go after a certain local market, maybe biologic, biosimilar or something like that, where we'll continue to manufacture in one. And not putting it past, there are some companies that have decided while they're building drugs to market as a biologics manufacturer, at the same time, people are going after each other's competitors as biosimilar. So these facilities can be repurposed for biosimilar purposes. So it gives you a lot of capability to utilize in a lot of flexible ways and also local regional ways. You can transplant whatever you want, wherever you want in a very simple way. So that's some of the ways that things are being done now. This is a more detailed view of what we showed before about how the time to market when you have to build. CMOs are are notoriously flexible. They have to be. Otherwise, they can't manufacture in the way that they need to meet their clients' demands. So they might be manufacturing anywhere within these. So CMO infrastructure with single-use and hybrid is really a powerful tool. Single-use in particular for contamination reasons is a very desirable technology for contract manufacturing organizations. They can reduce contamination risks and make it really easy to repurpose as they get clients in and out. When it comes to someone in the industry where they're actually trying to build, we already talked about the idea of conventional stainless steel. Somewhere in the phase 2 time you have to start versus versus the single-use concepts, which might be able to delay in phase 3, and we showed that already a little bit. So this is just a little bit different view overlaying CMO desires and such. So it's a really unique way of providing flexibility to your manufacturing capability and capacity in an organization. And it's happening. This is just, I'll end with this view of our slide, a slide that shows where we've seen our facilities being installed, our Flex Factory solution. We call our Flex Factory when we put the unit operations into an existing facility. And Cubio is where we build a whole facility. I'm speaking because I can speak on GE standpoint. I can't speak for all other manufacturers, but you can see it's happening around the world now. So we're talking about these flexible manufacturing cells being put in around the world, and it's increasing on a regular basis. So hybrid is the way of the future. Single use is an enabling technology that's going to be able to enable both the hybrid and the single use facilities around the world. And again, I feel it's important to understand this technology and the advantages that it brings forward. I think it's going to help drive the market and the industry for the next 10 years. So I appreciate your time, and I look forward to answering any questions that we can do to help clarify your minds on any of the topics we've covered.
Well, thank you, Adam. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send in your questions via the questions tab located directly below your webinar screen. And now for our first question. How does a single-use system reduce contamination risk? Sourav, would you like to answer that first?
Sure. So, and I think I mentioned during the slide presentation that the single-use systems do not have to rely on CIP and SIP to be working properly. They come pre-sterilized, and the bags are thrown away after each use. So, because of that, we see a measurable change in the level of contaminations, the number of contamination events that we typically have to deal with if we were operating a stainless steel plant. So that's primarily the reason for higher success rate in a manufacturing facility.
Thank you. Our next question is, what design aspects are important for a clinical manufacturing facility in comparison to a commercial facility for routine production? Sourav, would you like to take this again?
Yeah, so clinical manufacturing facilities are for advancing the pipeline, and by nature, by definition, they have to be flexible. They have to be able to accept different types of process unit operation sequences, different types of process technologies. Process technologies are evolving at a very fast pace, and you know, for a manufacturer, we always want to evaluate at least and implement, in many cases, new technologies that give us that operational edge. So the clinical manufacturing facilities are the test bed for trying out those types of technologies, and that's primarily why the clinical facilities have to be more flexible. In the commercial world, the process is well defined. Every change is a change control. Some have regulatory implications. So changes come fewer and far between. So they can run the same process for years and years.
Thank you. Our next question is, I have seen single-use bioreactors up to 2,000 liters, but I haven't seen chromatography systems at large scale. What are the largest single-use chromatography systems currently available? Adam, would you like to take this?
Yes, Stephen, I could take that. We've been seeing a trend with the idea of single-use. There's smaller columns that are typically being used, but we've also been seeing the idea that customers want to utilize their existing larger columns and larger processes and try to move more towards single-use for the cleanability aspect that Sourav had mentioned. So we're seeing a trend to want to have larger systems in single-use design. I know of what GE can offer today is a single-use chromatography system where the entire wet part flow path gets replaced. Up to 58 liters per minute, so about 3,500 liters per hour are possible at this point, which are capable of managing a column of 1 to 1.2 meters. So it's a newer products that we offer, but that is now large-scale single-use chromatography is possible at those. The flow kits are now capable of 32. They're effectively 3/4 and 1-inch flow kits, so 32 liters per minute and 58 liters per minute. So it's capable of being quite large now.
Thank you. And on the back of that, we have another question, which is, I see pre-packed process scale chromatography columns available from several vendors. Are people using these for single batches or many batches?
Well, I can take that one too. So it's an interesting question because the idea of single-use columns, we call it single-use, but when you talk about pre-packed columns, it might be plastic. They're not really single-use. What we're finding is people are using them for a variety of different methodologies. In many cases, what they might use them for are single batches, if you're kind of in a CMO world. So you would buy a pre-packed column and you would utilize it for that campaign, if you will. In some cases, if it's a company that's dedicated on a drug, we'll find that they're buying the pre-packed columns and utilizing just as traditional columns, except instead of packing them themselves, they are utilizing the pre-packed columns. And one of the problems that you walk into is you question how stable the column is because it's pre-packed and shipped around the world. Is it truly going to be stable when we receive it? I know from a GE standpoint, we do a great deal of validation on that, where we put it through a whole bunch of different equivalent shocks and temperature variation and such to simulate the shipping around the world and the temperatures and conditions it might apply to. So that allows it to be stable. So what we're finding is, again, depending on the application, very few people are using for a single run. Most people are using for either for a campaign or for if they store that column and utilize it on a certain type of drug when they make that drug on different campaigns as they come forward until the capacity of the resin is utilized up fully. So I think that's the best way to approach that answer.
Thank you. Our next question is, how can you repurpose QBio plants? Adam, would you like to take this?
Yeah, I guess since I mentioned QBio, I should probably take that. So QBio is a facility with flex factories built into it. So you put the equipment into pre-configured modular facilities. So by the very nature of it, we would probably deliver that to your design and your specifications up front. But the modular nature of the equipment allows for very simple reconfiguration because the connective technologies are often flex hose type of connective technologies. They can use sterile connective connectors, so you can actually easily reconfigure the hosing between them. So it's a very simple thing to roll equipment around, put into a new configuration. The automation can support the different changes because of the fact that the unit ops are actually predefined as kind of standalone, but can be connected into one another. And by doing so, you can reconfigure very easily. You roll things into a different configuration, reconnect the technologies, and you basically redesigned your facility. And the only thing that you'd be looking at is what buffers are you going to produce behind the wall, or what totes are you going to be rolling up to feed the room as you feed it through the wall, as Sourav has shown. So it's a very easy thing to reconfigure because of the modularity and maneuverability of these components.
Well, thank you for that, and thank you, audience, for your questions. Unfortunately, that's all we have time for. But before I finish the webinar, I would like to ask our presenters, that's Sourav and Adam, if they have any closing remarks or any final statements.
Okay, I can go first. I think this webinar was actually very good. We saw an existing facility that was built from an existing shell space, how it was set up and designed and validated. And then we had some great statistics and also example from GE Healthcare on all of the evolving technologies and business aspects that are becoming more and more apparent. And I look forward to the worksheet and advances in the single-use technologies.
Thanks, Sourav. And to echo that, I think that the job that Sourav and his team did to put their facility together was exemplary. I mean, they looked at their needs and they looked at the technologies and put together the right technology for their application. And I think that's the key when you put these ideas together of what do we need. You have to open your mind, look at the technologies that exist, and put the right technology for you. Because much like anything else in life, if it's wrong for your needs, it's going to be wrong. What makes it right is it matches what your requirements and your needs are. So I hope you had a chance to see what we interpret as the future and some of the ways that the future is going to be met. But in no means do we know the answers. It's going to be an exciting ride, just like it has been for the last 20 years. So we look forward to working with you. I invite you to check back on the GE website when this tool comes live and is available to you. I'd love you to play around with it and kind of get some ideas of what we feel the investments associated with single-use versus stainless steel are. And thank you for your time with Bioprocess Asia's support to give this webinar. And thanks, Sourav, as well for presenting with me.
And I would also like to take this opportunity to thank our presenters, once again, Dr. Sourav Kundu and Adam Kaletski, for sharing their knowledge with us. I would like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. And if you are watching this on demand and you have any questions, please email them straight over to me at stephen.edwards@biopharma-asia.com. Thank you, everyone, and have a great day.